forward.
Okay, thank you. Hello, thank you for inviting me, for the organizers of this symposium. My name is Michael Edwards. I am founder and CEO of Bioinfo Solutions. We are a data analytics company based out of Parker, Colorado. I used to be an assistant professor here for a number of years, but I really got sick of the commute, writing grants that didn't get funded, all-day faculty meetings, and just following other people's rules in general. So about 2 years ago, I formed my own company, and I like doing science, and this is what I really want to do. So as a bioinformatician, I kind of see myself more as a data wrangler than I do a number cruncher. That I consider all this information out there as pixels, and that if you consider your average flat screen TV, it has almost 2,000 pixels in it. These are colored boxes. And that when you watch your TV, do you see all these colored boxes when you watch your TV? No, of course not. Our minds puts all this information together to generate pictures, right? And we laugh and we cry at these pictures, but in essence, all we're doing is looking at colored boxes. So this is what my goal is with bioinformatics. How do I take all this information together and put it into a picture that my collaborators or my clients can understand? And right now, it's a great time to be a bioinformatician because there's lots and lots of pixels out there. This is a screenshot of Geo. I don't know if most of you are familiar with this, but this is where we're depositing the majority of our omic data. And right now, there are approximately 100,000 different studies in Geo, corresponding to 2.5 million different samples. There's gold in them there hills. And all you have to do, and what my company specializes in, is going into these databases and getting information that can benefit whatever system I'm looking at. To kind of demonstrate how my company does this, I'm going to talk about a meta-analysis I did on a rare kidney cancer. This was in conjunction with Illumina. There is a hackathon going on right now in Silicon Valley where they're actually looking at this particular rare form of kidney cancer. They bring together researchers, computer scientists, engineers, and they basically try to hack the disease. Illumina knew that I use their software quite a bit. They said, "Hey, do you mind taking a crack at this? We're donating our software for this hackathon. Maybe you can kind of look and see what you can find using our software." So I did. The type of rare kidney cancer is in the red box. It's papillary kidney cancer. About 8% of all people diagnosed with kidney cancer have the papillary form. The numbers here are a little too big. There's two main forms of papillary kidney cancer. There's a type 1 that's not as aggressive, a lot easier to treat. And then there's this type 2 that looks very different under the microscope, very difficult to treat, poor survival outcomes. And right now, all cancer researchers, oncologists, treat both these cancers the same way. So Illumina has a huge database. Obviously, they sequence 95% of everything on this planet. If you know somebody that works at Illumina, make friends with them. They're going to rule the planet at some point. But I got with Jim Flynn and Joe Delaney over at Illumina. We basically went through their database and tried to collect as many papillary renal cell carcinoma databases as we could find. And what we did was found 8 different human gene expression studies comprising 17 different comparisons. And these are given here along with their associated geo numbers. The majority of these comparisons are papillary renal cell carcinoma to normal tissue or normal adjacent. But in these last 3, we're actually comparing different forms of the cancer to itself. And in particular, the last 2 is the more aggressive forms, type 2 and poor survival, versus type 1 and excellent survival. Actually, this should be changed. I'm sorry. And also given here are the number of test subjects they used in those particular studies, the number of control subjects, and the amount of genes they found differentially expressed based on this criteria down below. We can actually visualize all those genes as it correlates to the expression. Again, this is the correlation engine platform in Illumina. Represented here are the different genes differentially expressed in those studies. A red bar would signify that that gene went up in that particular comparison, while a green color or green bar would mean that gene went down. The relative height of those bars represent the magnitude of that fold change. And what they are actually ranked based on the sum of this, the normalized fold change across all the different comparisons. And we can look here. What amazes me is, you know, if you've got a bunch of eyes looking at the same thing, they're going to see a lot in common. And that's what I love about this analysis. I don't have to trust just one group. I can look at all of this data. Given here are some, and you can see the consistency in it, which is really interesting. But what I want to point out here is if we're looking for a biomarker or a target for this particular papillary and renal cell carcinoma, we want something that is consistently down or up across all our different comparisons. And you can see here there's this one gene, dopa decarboxylase, or DDC. We can also use another Illumina program that called Cohort Analyzer that connects all this genomic data to clinical data. So this is survival. So this is the gene that I circled. This is DDC. And as you can see, with low expression, you get much worse survival than you do with high expression. But my specialty, and I don't like just looking at the genes itself. I like to take all these genes and put them together in kind of a one big picture, as I mentioned with the pixels, you know, and kind of look at the patterns of all these different, the expression of all these genes over all these different comparisons. So given here is a clustered heat map using Partek's genomic suite of the top 1,000 genes that went up and down respectively in this meta-analysis. So in all 17 different comparisons. If the color is blue, that means that gene went down in that comparison, and a yellow would mean that gene went up in that particular comparison. And if we start looking, and if we look at some of the, say, the type 2 versus the type 1s, we can notice differences in expression patterns of these genes, and clusters start coming out. In particular, we can look at this one here, circled in the red, that these genes seem to be upregulated in the type 2, the more severe types, compared to the type 1. And we can take a closer look at those. We can zoom in. Here's a cluster within that, showing the relative expression over all the different conditions that we looked at. We can now take these 130 genes. So basically, my theory is this is where I'm trying to get at. That I think trusting what some experts said 20 years ago about a pathway that may or may not be relevant to you, or may not even be accurate anymore, you know, I think is futile. That I think the true signaling pathways, you know, are in these kind of clusters, these genes that behave similarly. So we can take that and throw it through a program, a networking program called String. This is from the European Union. I should say this is, again, free program, which I really like. You don't see a lot of freeware out there that's very good, and I don't have $12,000 to spend on networking software, so I love following the Kaijin guy. But, what's that? Oh. Yeah, no kidding. Now, after they got taken over by Kaijin, I don't get any free licenses anymore. But anyway, you can throw these in, and what you can see is basically a mess, this mess of connectivity. And every line in this graph represents evidence of connection between these genes. So again, all these genes behave as similarly across all these different conditions. They're probably interacting together. And if we overlay different biological groups over this, we can see that the majority of these genes are cell cycle type genes, and they're also located in the nucleus. And one in particular, we can start pulling out targets from this. One of these that I like is topoisomerase 2A. It had the most connections out of any genes in this particular cluster. It also had the highest cumulative fold change out of any of the other genes in the cluster. And again, if we look at go to a cohort analyzer, look at, is there any clinical relevance? You can see that high expression of topoisomerase 2 correlates to a worse outcome in survivability. But what I really like to do is what we can now do is take all that cluster of genes that we've collected basically and throw it back into the original software. So this is throwing it back into correlation engine. And just like the previous speaker said, there's a lot of value taking your list and comparing it to all the other studies that are out there. And so that's exactly what I did. We can take those genes, and basically we were looking for similar patterns of expression. We could see that, obviously we're getting a lot of papillary renal cell carcinoma studies that come up, but we're also seeing breast cancer. In particular, our particular panel of genes behaves quite a lot like ductal carcinoma in breast cancer. So this is good information that, say there's a therapy that works in ductal cancer, there's a good chance it'll work on mine. You can also get other different aspects, cells that are more, breast cancer cells that are ER-negative are more similar to our cluster of genes. And this makes sense if you understand anything about kidney cancer. The men are twice as likely to get this disease than women, so maybe there's an androgen component in this. And also, the genes that we are looking at tend to be increased or upregulated in more severe forms of this particular breast cancer. So again, this is giving them more evidence that we're on the right path. And what's really cool, and I hope there's some NDs in here, but you can use all this information to look at, obviously Illumina has a huge chemical database. We can look at to see if there's any chemicals out there that affect our genes of interest. And if we look here, the first three were kind of false flags, but there is one, patinib, actually comes up and affects the majority of the genes that we're finding in this cluster. Again, these are genes that are upregulated in the more severe forms of papillary renal cell carcinoma. So again, potential therapies that we might be able to get to using this software. Deep down, I mean, if you get anything out of my talk, what I want everybody to understand is you don't need a PhD, you don't need machine learning, a fancy algorithm to see patterns, right? If you take all of this information and you just put it together and look at it, you can start seeing different patterns and start getting more information out of whatever you're looking at. And that's kind of what my company does, is we specialize in leveraging all the millions and millions of dollars worth of research information out there and applying that to your system so that you can get more out of your research. You can reach me. Some of the ways to, if you're interested in potentially pursuing this line of bioinformatics inquiry, I have a web page, Bioinfo Solutions. The website's bioinfosolutions.com. I also have a YouTube page. Please visit it. My 14-year-old daughter has 5 times the subscribers I do to her channel. So please, click on it so you like it. I actually go through, I have a whole series of videos where I talk about this rare cancer. I go from the beginning so you can see exactly what I did and how I came up with my lists and my conclusions. I'm also on Facebook. You got to be everywhere. I refuse to get on Twitter, so don't even look for me there. So it's a nightmare. And I'm even on Spotify. So check me out on Spotify. One of these days, I'm hoping like somebody comes to me after one of these talks and goes, I think your science is great, but I really like your playlist that you put together. What's your favorite Beta Band, album? But so I will take questions. Thank you very much. Any questions? Yes.
So you guys, if my understanding is correct, you guys take a bunch of different papers and you try to use the data from them to come up with like different networking with whatever they're studying. Is that right?
Say it one more time.
Sorry. You take different papers that are already published and you use their data and you sort of group them based off of what they're studying. Is that correct?
No, I don't have to trust what they say. I hate to, spoiler alert, some scientists lie. So I would rather, interpret their own data. So what I'm doing is I'm pulling out their gene list is what I am doing, not the text surrounding their study. So what I'm doing is comparing my gene list to all the gene lists that are out there.
So my question is, how do you, I guess, reconcile differences in methods or maybe differences in cohorts throughout different time periods?
Totally. And I mean, that's part of the, that's the good thing is, well, number one is, so Illumina will normalize all the fold change information. So in RNA-seq is a lot more sensitive than, say, array studies. So you're going to get larger fold change information in the RNA-seq. So what they've done is the highest fold change you can get is 100, and then they just basically make it a linear scale to 0. What I've also found is if you compare different species like mouse and human, you see a lot of agreement as well. The nice thing about this is we can pick and choose which comparisons that we make, and it's completely fluid the way we go with that. Yes?
Do you do anything to account for publication bias?
I think by taking as many... So, does everybody know who Nate Silver is? He was 538. Well, basically, he was famous because he predicted basically every major election in 2012 in this country. And he didn't take any surveys himself. What he did, and he took all the surveys out there. Some were obviously biased left, some were obviously biased right. But when he took the middle of all those surveys, it was pretty much the exact results of the election. And that's kind of what I do with all this biological data, is if I'm taking from so many groups, one group could be terrible, one group might be really good, but the average of that, I'm going to probably get the right answer. So I'm not really worried about, potential publications interfering with that.
And you can also see the outliers. When you align all the studies, you can see
Totally.
If you have some studies that are not...
Right. When I put them all together, if 16 out of my 17 look pretty similar and one is just completely off, and I do see that, you can always throw that one away.
All right. Thank you very much.
Thank you.